We are now going to look at the developmental mechanisms that can produce large anatomical variations. We will be using those three principles of Evo Devo, the small toolbox, enhancer modularity, and gene duplication, to look at the mechanisms by which changes in gene expression can give rise to anatomical variations. What we find is evidence for what Francois Jacob called evolutionary tinkering. Nature rarely makes new genes, but repurposes existing ones. Fortunately, as Wallace Arthur has pointed out, there are only four major ways that this tinkering can take place. The first way is to change the place of gene expression. This is sometimes called heterotopy. In other words, the cells that express the gene can be different between species. The second way is to change the time of gene expression. This is sometimes called heterochrony. The third mechanism is changing the amount of gene expression, heterometry. And the fourth mechanism is to change the kind of gene being expressed, to actually change the protein sequence. This is called heterotypy. Now, heterotopy, changing the cell types that express the gene, can explain, for instance, how the duck got its webbed feet. Most land-dwelling vertebrates start off with webbed feet in the embryo. Our limb bud is webbed, but as you can see, the chick hind limb has freestanding digits like ours. The duck hind limb has webbed feet. During development, there is cell death in the webbing of the chicken limb, just like in our limbs. And that's shown by the red dots on the chicken webbing. However, the duck has very little cell death in its webbing. The cell death is caused by signals from paracrine factors called BMPs. And the BMPs here are expressed in both the chick and in the duck hind limb webbing. So what's the difference? The difference turns out to be whether or not inhibitors of BMPs are made in the webbing of the limbs. In the duck hind limb, the gene for gremlin is being expressed, and gremlin inhibits BMPs, so that the BMPs are not allowed to signal for cell death in the duck hind limb. In the chick hind limb, gremlin and other inhibitors of BMPs are not being made. Those genes are not being expressed, and so BMP is free to cause the cell death in the webbing. You can show this experimentally. You can take a bead and coat it with a salt solution, a saline solution, and put that in the webbing of a chick embryo. The webbing is not saved, the BMPs destroy the webbing, and you get freestanding digits. However, if you coat the bead with gremlin protein, such that gremlin protein is dispatched throughout the webbing, what happens is you get a duck-like region of the limb, such that the webbing is retained. This is also seen in the bat wing. So here the forelimb keeps its webbing. The bat limb is an extension of the fingers of the bat and the retention of the webbing between the fingers. All mammalian embryos start off with webbed forelimbs, but the bat is somehow able to keep the webbing there. And you only want the webbing in the forelimbs of the bat, not in the hind limbs of the bat. The hind limbs of the bat are free digits such that it can perch. What happens is a BMP protein, BMP7, signals the cells to die in the webbing of the bat hind limb and the bat forelimb. But the bat forelimb webbing expresses the genes for gremlin and FGF8, two factors that inhibit BMP signaling and thereby allow the webbing to be retained in the forelimb of the bat, but not in the hind limb. Heterotopies, changes in the cells expressing certain genes, are very important in making evolutionary change. One of the new structures formed by heterotopy is the turtle shell. The carapace of the turtle is formed by ribs that go into the back dermis. Now, most ribs go ventrally, forming a rib cage meeting at the sternum, but not in the turtle. Unlike any other animal, the turtle's ribs migrate laterally and they actually enter the dorsal dermis. It's believed that this migration into the dorsal dermis is occasioned by the expression of FGF10 in certain cells of the dermis. FGF10 attracts the rib precursors into the dermis, and that's where they form their rib, not ventrally, but laterally. Once they are in the dermis, they then can signal to the skin to form the rest of the bony plate of the carapace of the turtle shell. So what makes a turtle a turtle? Its ribs. Why do the ribs go in a new direction? They are attracted there by a paracrine factor that normally is not expressed in the dorsal dermis. So heterotopies, changes in the place of gene expression, is a very important mechanism to getting evolutionary novel structures. Another way of doing this is heterochrony. For instance, how does the dolphin get its flipper? Well, FGF8, which we just mentioned as an inhibitor of BMPs, also functions as a mitogen. It functions to keep cells in a state of proliferation. Normally, after a certain period of time, the FGF8 gene is turned off and limb development stops. However, in the dolphin, FGF8 signaling is retained. The gene for FGF8 remains active, and as a result, you get these long fingers, which are characteristic of the dolphin flipper. Changing the timing of gene expression is also seen in the formation of snake ribs. Some snakes can have up to 300, sometimes even 500 ribs. How do they get so many ribs? They actually shorten the time it needs to make each rib from each somite. And as a result, you get a large number of ribs made at any given time. One of the most important heterochronies might be human brain growth. The human brain keeps growing at the fetal rate for over 2 years. In other words, we're still making new neurons. In other mammals, including other primates such as the chimpanzees, they do not develop their brain continuing the fetal growth rate. That's something that humans do. And what one finds is that there are numerous genes that appear to be active in allowing humans to keep a rapid rate of neuronal growth after birth. Many of these genes are inhibitors of inhibitors. For instance, the GAD45G gene. This encodes a protein that is an inhibitor of neural growth. Humans have lost the neural specific enhancer for this gene, thereby allowing this gene to be expressed in other organs, but not in the human brain. Humans also, as mentioned earlier, have an imperfect duplication of the SRGAP2 gene, such that one of the duplicates actually encodes an inhibitor of the original gene. The original SRGAP2 gene encodes an inhibitor of neural growth, and the copy, the imperfect copy, actually inhibits that inhibitor. And here we see a very important principle of developmental biology. Activation is often the inhibition of an inhibitor. Now, heterometry, the changing the amount of gene expression. This has been seen to be critically important in the evolution of those finches that Darwin observed. There are broad-beaked finches and there are finches with narrow beaks. Those finches with broad beaks have an embryo with a craniofacial region that expresses large amounts of BMP4. Now, BMP4 we saw earlier was involved in cell death, but in the facial region, BMP4 activates mitosis. And this is another principle of developmental biology. What a protein does depends on the region of the embryo where it is being expressed. So BMP in the webbing of the limb causes cell death. BMP in the craniofacial mesenchyme causes cell proliferation. This is what it does in Darwin's finches. The more BMP4 that's being expressed, the broader the beak. This can also be seen experimentally. You could take active BMP4 genes and add them to the beak primordium of embryonic chicks. And what does it do? It actually broadens their beaks by causing more cell division. So that's heterometry. So the next way that you could get anatomical change during development is heterotypy, changing the identity of the gene being expressed. This actually can help us explain why insects only have 6 legs. Most arthropods have a lot more. If you think about a seafood platter with lobsters and shrimp and crab, they have lots of legs. Spiders have lots of legs. Millipedes and centipedes are known for having lots of legs. Why do insects only have 6 legs? In the group that became insects, there was a mutation in a gene called ultrabithorax. Ultrabithorax acquired new amino acids in the protein, and these amino acids were able to repress the distalis gene from being active. Distalis is a gene that activates other genes that help form the limb. If it is repressed, the limb doesn't form. Ultrabithorax, which is active in the insect abdomen, is able to inhibit distalis, and as such, the abdominal limbs of the insect fail to properly form. So, there are 4 ways of tinkering to get large anatomical variations. There's heterotopy, changing the place of gene expression. Heterochrony, changing the time of gene expression. Heterometry, changing the amount of gene expression. And heterotypy, changing the identity of the gene being expressed. Gavin de Beer, writing in 1953 in a book called Embryos and Ancestors, said, "But since phylogeny, that is evolution, is but the result of modified ontogeny, development, there is the possibility of a causal analytic study of present evolution in an experimental study of the variability in genetics of ontogenetic processes." Finally, he said, "It may be possible that freed from the trammels and fetters which have so long confined thought, the whole of the animal kingdom may appear in a new light, more homogeneous and compact than had been imagined, and with the gaps between its major groups less formidable and perhaps even bridgeable. Evolutionary developmental biology is making those gaps less formidable and is building the bridges that unite them."